Yeah, we got several things running. On the ancient DNA side, we got several samples that have been soaking that we need to get cleaned up and maybe prep for sequencing, just depending on if we see DNA in them. So that's exciting. We're going to have some meetings this week about training the Basecalling model to identify the signals of ancient DNA. And so we got a call today about that, so I'm excited about that. And we'll see if we can make some progress on that front. We're also looking to add some new equipment over the next month or two, so we're doing a little bit of searching to find the right computer and everything to work with all that. So we got several things moving on the kind of computational front as well. And then kind of on the experimental side, looking at topoisomerase stuff, we're writing a paper on AlphaFold and using it to look at the structures of topo. So it's kind of a follow-up to a paper that was published last year by us. And so I'm looking to kind of expand that work a little bit. Last week we successfully fluorescently labeled topo, and so we're going to label some more this week and try to start testing out whether we can use this fluorescently labeled topo to detect DNA binding in an anisotropy experiment. So I'm really excited about that. And then on the kind of the front of looking at topoisomerase and how it works, we're working to purify 5 mutants. So last week we purified some normal or what we call wild type enzyme, and this week hopefully we'll purify the first mutant out of the 5. We've grown up 2 so far. We got the rest growing. So sometime over the next 2 weeks we should have all of those grown and harvested, and then we'll be purifying them over the next week or 2. And then game on. And we just got to start running the experiments to test them, to see how they work, what they do, if they work or don't work. We'll test them with the binding assay, with anisotropy, if that works. So we got a lot of cool things coming. So we'll see. This week will be a busy week. We only got 4 days, but we're going to pack a lot in those 4 days. So.
Adding some topo into the mastering, so I'm basically doing a DNA cleavage assay. We're working on a DNA relaxation assay. Dr. DeWeese did one earlier this morning, and then I just did one myself, and I'm about to both the girls to run both of their gels. But the end goal right now is just practicing to make sure we can get a good relaxation gel for when we start our mutants. Because once we start our mutants, we're going to need real results.
Finally gotten the bones, the DNA out, and it's time to centrifuge it.
And then do that with your other 2 and tighten it down on there. We have to hold it. 5,000 RPM. It's usually
2 hours later.
It is day 3 of summer research with biology department, and we have
Madeline.
and we have McKenna, who's joining us on the ancient DNA research. She was on her side, but today she is on my side. So she's basically a traitor.
What did he say?
Some topo. What are y'all doing today?
So today we're aliquoting topo, which means we're taking a big tube. We have about, I think, 3 liters of it, and we're putting it into tubes of 10 microliters and 20 microliters so that we can use it in our assays.
60 to 70. So it's good.
So buon po. Ja pilun jan. Benza.
I am putting some samples into this plate, and then we will read them with the spectrophotometer, which ideally will tell us which wavelengths of light they absorb the most. And determine the concentration of dye in the samples, which is important for later when we want to label proteins. That way we know how much we have, so we can do more specific ratios of dye and protein.
Finally cleaned up some of these samples, these fossils, and we are getting ready to spec it.
Basically what we did this week is preparing this new topo. So we had our old topo wild type, and that's what we've been using to kind of like run relax and DNA cleavage and just these type of assays just to kind of train us to be able to do this before we start on our mutant topo. And another thing that we did was also we aliquoted new topo. So basically aliquot is just pulling from this bigger tube into smaller tubes and putting it in the freezer. And what we did yesterday was we pulled some new topo and some whole topo and kind of compared it. And instead of like the normal gels that we kind of see in the little box, we did this thing called a protein gel. And that's kind of like standing up, it's kind of weird. But basically what that does is that we kind of see the different proteins in each of the tube samples. And what I did yesterday was that you can see on the actual thing. So for here, this is the old topo. Different like wells is like 1 microliter of topo, 2 microliters, and 3 microliters. This is just to kind of see the different concentrations and the variations, if any. And this is the new topo, so that's again the same thing. We put 5 microliters of dye and whatever remaining to get to 15 microliters in total of DI water, so deionized water. And what we did see, ideally what we like to see is this. So there are no bands, everything's clean. Basically what that's saying is that there's only topo in those tubes. But this one is not as clean because there are different bands over here. So basically what that's saying is that topo is not alone. There are other proteins.
We've been doing a lot of testing with our relax assays. We figured out what went wrong with our gels. We were using the wrong ingredient in the actual mold of our gel. We've kind of perfected relax. Now we're working on a enzyme titration, which is we dilute our topo we've gone from 1 to 1/40, and we're testing how strong it is. So we've also prepped wild type and aliquoted it, so we have a bunch of extra topo to work with. And then we have gotten mutants 2, 3, and 4 ready to prep and ready to put in little tubes that we can use in our tests to see how it works, see how strong it is. So next week's probably gonna be big, just getting things done, experiments and all that, but
on finishing up research that I've been working on since last summer. The big goal that we're looking forward to is publishing a paper about it, so that's been really fun. Basically, we study this enzyme called Topoisomerase. That's kind of what it looks like there. There's this program that can essentially generate folds or how it predicts the protein's going to be. And this is how it thinks that topoisomerase 2 beta, one of the proteins that untangle our DNA, would exist in nature. If you look, I've color-coded it. See this yellow region is what's called the CTD. It's intrinsically disordered. Basically, it's part of the protein that doesn't fold well. It's kind of pretty chaotic, that's how you can think of it. However, we figured out that we can add certain mutations. It can go from this jumbled up mess to something that AlphaFold predicts, hey, it's a little bit more compact. So it's interesting. If we add more DNA, we can see it predicts that it'll fold differently. So basically, I'm doing a lot of computational analysis of it, seeing how it changes, looking at the structures. For example, sometimes proteins fold in little helices or like little turns. Sometimes they form in like little loops, and we see that more mutations can influence that. And so it's really interesting. Right now, I'm just working on getting the paper done. We have a draft here, have a bunch of fun figures, but yeah, that's what I'm working on now.
Our goal for this summer was mostly to train a new Basecalling model that would be able to differentiate between, is it thymine and uracil? And so we've been working throughout the past couple of weeks towards that goal, and we have made a lot of progress. And we have decided to name our model. And so we had a few different names going through for a while. We had we had the wahoo, the cusk, the barracuda, and the chimera, or the ghost shark. We ended up going with ghost shark as the definite name, and so I guess you could use chimera if you want to be more like scientific about it. And so this is an image of what the ghost shark looks like. I don't know how well that will come up on the video, but so ghost shark is what we ended up going with, and so we're going to kind of walk you through what some of that process looked like.
We trained it fully intending for it to fail, and it did fail at first, but then we made some modifications to it, and now it actually works pretty well. When we inserted some test data into it for control thymine and for actual test uracil, we found that it could call thymine as thymine 96% of the time, and could call uracil as uracil 83% of the time. And we only had the false negative or the false positive scenarios at 4% and 17%. And that's being able to extract over 80% of uracil compared to what we had before, which was closer to 0%. That's actually a massive improvement.
Our struggles were just like, we didn't know why it kept failing, and then one run just worked.
Hey, week 2 has been exciting. We've got some great work moving. Our topoisomerase assays are going well as far as the students getting trained and getting going on those. We're growing some more topo. We're testing some that we just purified. And then hopefully next week we'll start purifying the mutants, which is exciting. So we'll have 3 that we can purify. So we'll start working our way through those over the next weeks.
Oh yeah, you're right.
So tell me what you're doing.
No, this is actually overwhelming.
Yeah, you see? Let it be known, Philly doesn't like to be in front of the camera, but he loves to shove it in everybody else's faces.
To you, happy birthday to you, happy birthday dear Melvin, happy birthday to you.
Okay, I was like, is this what I was like, did y'all prank me with this?
No.
And so, you know, it's like the glass coming over here. Billy was like, Sarah, you drive with 2 hands.